Hello everybody, Fritz here. In today's video, we're going to go over your operons. Now the two operons that we're going to look at today are going to be your Lac Operon as well as your Tryptophan Operon, which can be abbreviated as TRP. The Lac Operon is what we would consider an induced type, while the Tryptophan Operon is considered a repressed type. And both operons have similar components. Firstly, we have a regulator, followed by a promoter, an operator, and a gene of interest. And that is common for the Tryptophan Operon as well. Let's first look at the Lac Operon. Under normal circumstances, the Lac Operon will have the regulator make a repressor. This repressor will then bind to the operator. And because the repressor is bound to the operator, RNA polymerase is prevented from traveling down this segment to transcribe the gene of interest, which is lactase. Lactase is the enzyme that will break down lactose into glucose and galactose. However, if the bacteria has enough glucose in its environment, it does not want to waste energy and break down lactose because it's just going to get glucose that it can use for energy. So under normal circumstances of high glucose, we will not have this operon on because it wastes energy. However, when we have a situation in which glucose is low and lactose is high, the bacteria will want to break down that lactose with lactase in order to give us that glucose molecule because, once again, lactose is a disaccharide of glucose and galactose. When those two prerequisites are met, the bacteria will create an inducer, which is allolactose. Allolactose will bind to the repressor, preventing it from binding to the operator. And then we are allowed to have RNA polymerase travel down the entire segment and transcribe lactase. And this process in which the inducer as well as this segment of the operator competing for the same site on the repressor is called competitive inhibition. This means that the binding of the inducer or this segment of the operator will directly have a negative impact on the opposite component. So the inducer bound to the repressor will allow RNA polymerase to travel downward, but the binding of the operator to the repressor will prevent RNA polymerase from moving forward. So that's why they are both competing and inhibiting one another. Therefore, competitive inhibition. Now, looking at your Tryptophan Operon, RNA polymerase under normal circumstances is always allowed to travel down the segment and transcribe the gene of interest, which in this case is going to be your tryptophan. Tryptophan is a protein that the bacteria can use for certain biochemical processes. However, if there is too much of it, then it's a waste of energy to keep on making tryptophan. So how do we turn this off? Well, the regulator segment, same as the regulator segment over here, produces a repressor. However, this repressor is incomplete. It cannot fully bind to the operator and stop RNA polymerase from moving forward. Therefore, the corepressor is produced, nonetheless tryptophan, and acts as a corepressor. These two will bind together in order to bind to the operator, stopping RNA polymerase from moving forward and transcribing more tryptophan. And this action in which the final end product acts as an inhibitor is called negative feedback, because the actual end product is telling the system to stop making any more tryptophan because you have enough. Enough so much that it acts as a corepressor to turn this off. Now, you will be expected to come test day to remember which one of these is the induced type and which one is the repressed type. What helped me out, and I hope this helps you guys out too, is that the letters L and I are closer together alphabetically, and then the letters T and R are closer together in the alphabet as well. Therefore, L and I pair together. So the Lac Operon is induced. And because T and R are closer together in the alphabet, tryptophan is repressed. But you will also want to know which one of these acts on competitive inhibition and which one acts on negative feedback. And here are some additional questions that you can be expected to be asked on test day. And as always, please feel free to leave those answers down in the comment section below, as well as any other questions that you might have. And don't forget to check the description for additional study materials. If you found this video helpful, please leave a like and subscribe, and I'll see all of you in the next one.